Just look at what we call amino acids. They are basically the building units, the building blocks. Okay, amino acids are the building blocks of proteins. Okay, and the proteins, we are saying they are large molecules that consist of several of these amino acids. But of course, we can have proteins that we can call, for example, dipeptides, meaning that you have two peptides that are connected to form these peptides. And they are basically existing as peptides, dipeptides, and that's how they are functioning. Okay, so you will see how these actually combine in this presentation. And let's start with this kind of scenario. So we have a boy who has been diagnosed with a genetic disorder where one amino acid in a protein is replaced with another. Can you explain how such a mutation could affect the function of the protein? And then can you draw and label the general structure of an amino acid? And can you differentiate between non-essential and essential amino acids? Can you give an example in each? And then can you describe the importance of amino acid sequence in a polypeptide? Now, we can answer some of these questions here right away. We already noted that amino acids, the sequence, that is the arrangement, which amino acids and which amino acid follow the other, that sequence determines which protein we are going to have. And therefore, if we remove one amino acid from a long chain of amino acid, that means that we're going to form a different protein. And when that happens, it's a problem with the mutation. So what mutation means is that you have a change either in the genes or in the structure of the chromosome. And sometimes these mutations are dangerous. That does not mean that you can not have mutations that are positive. Now the question is, how are these mutations in the genes or chromosome actually related to the proteins? The point is that it is from the genes, it is the genes that instruct the cell the type of amino acid to be made, and consequently the type of protein to be made. So that means that if there is a problem in the genes or in the chromosomes, then you're not going to have the right protein. And remember, the proteins are very many and they perform different functions. So for example, if you have a protein that is supposed to be used to form a particular enzyme, and that protein is not made, that means that that person will not have that enzyme. And that is going to be a problem. And that is a genetic disorder. It is inherited. It is an inheritable problem there. Okay, so that is how a synthesis of a particular protein is linked to the genes. So that therefore means that during the process of making proteins, it's very important to make sure that the sequence of these amino acids is actually followed. Okay, so we start and look at the other details. So an amino acid is a monomer of protein. So that means that a protein is actually a polymer where you have several amino acids combining. The combination of these amino acids is through what we term as condensation reaction. A condensation reaction gives out water. So what definitely happens is that you have the amino group reacting with the carboxyl group. Each amino acid has three components. Okay, you have the amino group and the carboxyl group, and then the side chain. Remember, that these are all connected to what we term as the central carbon. And we are saying that it is this R group, it is the R group that will be different in the different amino acids that we have. And so that means that the R group determines the characteristics that this amino acid will have, whether it will be a polar amino acid, whether it will be a non-polar amino acid. Okay, and of course, that polarity will influence the chemical reaction that this amino acid will have, and therefore the function. But remember, amino acids can be obtained from the diet that we eat, and we say those are essential amino acids. So if we eat, for example, meat, chicken, whether beef, fish, eggs, dairy products, cheese and milk, soya, and the rest, we can get essential amino acids from the diet. Okay, but again, the body can synthesize amino acids, and we term them as non-essential amino acids. Okay, and these can be synthesized from different sources: grains, legumes, nuts, and vegetables. Okay, so the R group we are saying is the side chain, and it can differ in different amino acids. We have over 20 amino acids, and they can be in that side chain, or the R group can actually be, it can be a hydrogen atom, okay? Or it can be a group, a complex compound that is connected there. So we can expect different chemical species replacing that R group, and they therefore making different amino acids. Okay. Now, we are saying that the R group can be hydrogen, just the hydrogen atom. It can be carboxyl. It can be hydroxyl. It can be amino groups. It can be a number of things. Now, if we have the R group as a long chain of hydrocarbon, then we could expect that amino acid is not highly reactive. Okay. Okay, it is not going to be highly reactive. But if you have a chemical species that is highly reactive, replacing the R group or placed in the place of R group, then you could expect that amino acid to be highly reactive. Okay, so if you have a long chain of hydrocarbons, then you cannot expect to have a polar hydrophilic amino acid. So if you have a long chain of hydrocarbons replacing that R group, or in the place of R group, you could expect that you have hydrophobic non-polar amino acid. Okay. So we move forward to scientists write these different amino acids. They will always not write the full name of these different amino acids. Remember, we talked about the 20 amino acids. Okay, so some of these amino acids can be represented by a single letter. For example, K can be for lysine. Okay, and just a single letter representing the amino acid lysine. R representing arginine, and then A representing alanine. These are basically, if you are in the field of biochemistry, these are codes that you can read and know. Okay, and then sometimes you can have three letters that are representing a single amino acid. For example, AL, A can represent alanine. Okay, so amino acids, we said that they are the monomers of proteins. That means that proteins are the polymers. That means that you have a combination of these different amino acids to form a protein. And we said that the protein can be different from another based on the sequence of these amino acids. And therefore, we could expect that you have bonds between these amino acids, and these are basically peptide bonds. Okay, so that means that proteins are like other organic compounds that we looked at. Carbohydrates, they are polymers. Lipids, again, polymers. Nucleic acids, these are also polymers. But also, we need to note that nucleic acids are actually macro molecules, bigger molecules. Okay, so the process of linking up is best to form a large molecule is what we call polymerization. But that linkage, when one monomer connects to the other monomer, that is what we term as condensation. The nature of that reaction is condensation because water is released. But breaking that molecule or that bond that has been formed is what we term as hydrolysis, because definitely water will be used in that process. So when you add water, that bond will be broken such that you have monomers from the polymer. Okay, so we are lastly saying here that the reactions are in chemical equilibrium. This means that it is possible to have hydrolysis taking place and then condensation again taking place. So you have forward and then backward. So these amino acids combine through peptide bonds, and usually the R group can influence the extent of folding and the nature of interaction with water. Okay, so what we are basically saying is that the side chain can interact through either hydrophobic interactions or through electrostatic attraction, ionic bonds and things like that. Or if that R group is hydrophilic, then you could expect interaction with water. If it is hydrophobic, then you expect it to basically fold, okay? Fold when put in aqueous situations. Okay, so the combination is such that you move from the nitro group to the carboxyl group. Okay, that is the direction. This part is the nitro group connects to the carboxyl group, and the nitro group connecting to the carboxyl group like that. And that connection is basically the peptide bond that we are talking about. Okay, if you have basically a straight chain of amino acids, then you could expect that it is very easy to bend or to fold this protein, and therefore there is some great degree of flexibility when you have single bonds in a particular protein that is basically linear. Okay, so the extent of the length of this chain can give us, we use different names to describe the extent of the length of this chain. Okay, so if we have just a few amino acids, that's a peptide. If you have very long amino acid chain, then that is a polypeptide. 50 and more, that is a polypeptide. If you have fewer than 50, that is oligopeptide. And then, of course, when we talk about proteins, these are very long protein molecules consisting of polypeptides. Okay, so polypeptides, basically when we talk about polypeptides, we're basically referring to protein molecules. Okay, and the proteins refer to the functional forms of these chains. Okay, so if it is an enzyme, that is a globular protein. If it is, for example, hemoglobin, that is a protein that is for transporting gases, and things like that. Different properties because of the fact that they're actually not the same. Okay, but for you to know the properties of these amino acids, still you need to relate these to the structure of the amino acids. Okay, so proteins are actually colorless and crystalline solids. They are soluble in water and insoluble in organic solvents. Okay, of course, the extent of solubility is what differs because of the different side chains that they have. And then they are amphoteric in nature, meaning that they can exhibit both basic and acidic properties due to the amino and carboxyl groups. They can exist as Zwitter ions at neutral pH, meaning that they can have both the positive and negative charges when they are in neutral pH. Neutral pH is around 7. So each amino acid has its own isoelectric point. That means that when it is electrically neutral. So always when thinking about the properties of amino acid, think about the chemical structure of these amino acids. Remember the 3 components. And the R group is the one which usually differs. So the R group can be occupied by a nonpolar or a polar group, and that will definitely influence the properties that these amino acids will always have.